Hello everyone, good evening. Welcome to the Santa Fe Institute public lecture series in our new venue, which is absolutely gorgeous. For those who are new to these events, I just remind you of why we do them. And there are three primary motivations for holding these public lectures. One, and I'll just read them out, is to promote conversation and debate around complexity science here in Santa Fe and through our videos online globally around the world. So it's rather evangelical. The second objective is really just to shine a spotlight on ideas and frameworks and people who we think are making a constructive contribution to rational debate in the world. And finally, really enriching the lives of people everywhere. So those are our objectives. And I should say again, this is a new partnership with Lensit for us, and we're just delighted. And I think we have 800 people at this event this evening. I wanted to acknowledge Thornburg for their continued support of the public lecture series. I'd like to be able to acknowledge many more people and companies. That's a hint, in case you were wondering. And so if any of you feel inclined to make a contribution to the Santa Fe Institute and our outreach activities, I'd encourage you to do so. I believe there are envelopes, very capacious envelopes, somewhere in the lobby. We're just delighted to be kicking off this series with tonight's incredible speaker, Karissa Sanbonmatsu, who's a principal investigator at Los Alamos National Lab. Just a little bit of background on Karissa. She became interested in chaos theory while studying physics at Columbia University in New York. She then went on to study nonlinear dynamics, in particular in astrophysics, in Boulder in Colorado. And then she took a rather circuitous route via New Mexico with an interest into the origin of life, and then currently epigenetics. She works on the bleeding edge of genomics, sort of investigating all of those mechanisms above and beyond the rather simple determinism of DNA, helping us to understand inheritance, individual variability, and our subtle responses to the environment. Life, it turns out, is far more complex than we ever thought, and that makes us at the Santa Fe Institute very happy. So please join me in welcoming Karissa.
I love you. When you're in a relationship, when do you say, I love you for the first time? When do you define the relationship? And when do you make the relationship Facebook official? Well, we all know the symptoms of love. The butterflies in your stomach, your hands getting clammy, so nervous you're terrified, but you can't wait for more. There's a lot going on when you fall in love. You thought it was just a bunch of heartache and money. Your brain goes through all kinds of changes. Your brain is flooded with oxytocin, the love chemical. Falling in love is physiological. On the other hand, since the time you could walk, you've heard stories about love from everyone. So is the ability to fall in love genetically programmed, or were you conditioned to fall in love? Well, we don't know. Well, what exactly do we mean by nature versus nurture? By nature, what we mean are things that are instinctual or hardwired into your DNA. So things like physiological traits that are heavily influenced by genetics. Some examples include migrating geese. So the idea when the geese are genetically programmed to know exactly when and where to go at what time of the year. Another thing that could be hardwired is your personality. And then finally, many things that are hardwired are studied in twin studies. Now, what do we mean by nurture? Well, these are more learned behaviors. So things like, for example, a toddler throwing a temper tantrum and always getting what they want. That's kind of a learned behavior. Also, maybe a dog learning to balance 3 oranges on its head, another learned behavior. And then also what we do in the lab doing molecular biology, that's a learned behavior as well. Now, there's a third class of phenomena that are not really nature and not nurture, but somewhere in between in this more nebulous area. And these include things like anxiety and stress, or stress disorders. There, it's not so clear if this was hardwired into your DNA, or was it a result of growing up in a very stressful environment, maybe. Another thing is laughter. Believe it or not, we don't really understand right now if laughter is something learned or is it genetically programmed. There are studies in primates showing that the ability to laugh is sort of taught to the younglings, but on the other hand, there are also arguments that it could be genetic as well. And then intelligence is another controversial subject, and then also the ability to fall in love, another. So this brings us to what we call epigenetics. And so epigenetics is sort of a layer above the DNA. And phenomenologically, it results in these kinds of traits that are sort of not nature and not nurture, but somewhere in between. It's sort of a layer of circuitry over the DNA that determines when and where genes are turned on and off. And an interesting metaphor for epigenetics is your smartphone. So if you think of your smartphone as like the DNA or the hardware of your smartphone, then the software or the app on your smartphone is like an epigenetic program. And then finally, the output of that app would be like the cell fate. So for example, if your app is something like Google Maps and you needed, suppose, like when you're coming to the Lensic tonight, you're trying to figure out exactly where to park and you wanted to look on Google Maps where to park, you use your Google Map app, and then the result of using the app is it gives you the directions to get here. And similarly, these cells have epigenetic programs that are sort of a layer of software on top of the DNA that can convert a cell into a blood cell or a brain cell or such as the embryo is developing and the egg is developing in the womb. Whoops, sorry. Okay, let me just back up a little. It's a little squirrely, the control here. Okay, so just to give you a basic overview of what DNA is. DNA is basically a long polymer or a long string-like molecule, and it's composed of millions of these chemical bases. You may have heard them as A, G, C, and T, or adenosine, guanine, cytosine, and thymine. And these millions of bases are organized in units called genes. And so one gene is basically one segment of DNA that codes for a protein, and those proteins are the things that do the actual work in your cells. And this is sort of the basic makeup of DNA. And then here is a 3D image of a DNA molecule. And this is magnified, millions of times. And so here we're looking at the length scale of 1 nanometer. And these are the chemical bases that I was talking to you about that make up the DNA. And nowadays, people are able to go to particle accelerators and use very powerful X-ray beams and blast DNA to get diffraction patterns and image DNA in atomic resolution. And it takes the shape of this famous double helix discovered by Watson and Crick in the mid-1900s. Now, there's sort of a packing problem that goes along with DNA. So there's enough DNA inside your body to wrap around the entire Earth several million times. So it's a lot of DNA. And so somehow all that DNA has to get packed inside your body in such an organized fashion that it can basically control everything that happens in your body. And the question is, how does nature solve that problem? Well, the answer is there are millions of chemical spools that wind up the DNA. So in each cell, there are thousands and thousands of these spools that sort of store the DNA. And then what epigenetics is, it's this circuitry that sort of unwinds and winds up the spools at just the right times and just the right moments. And so here are some other visualizations of the DNA wrapped around these spools. The jargon name for the spool is called a histone. And so here we're looking at a micrograph with a length scale of 0.1 microns. And you can see in 3 dimensions these red histone spools, and the DNA is in gray, and it's wound around those spools. So this is the DNA wrapped around the histones. And the amazing thing is that this intricate network of spools is highly, highly programmable. And it's what takes a fertilized egg all the way up to an embryo to an adult human. All those decisions that are made to differentiate cells into different kinds of cell types are made through controlling this network of spools or histones. And so this is essentially what epigenetics is. It's this layer above the DNA that allows the DNA to be programmed. So the DNA is organized into genes, and then the genes are wrapped around these spools, and when the spools unwind, then the DNA, then the genes can be turned on and off at the right moments. And this overall process is called epigenetics. There's something above genetics. And so again, if we go back to this analogy with the smartphone, just to hammer this home, the smartphone is the hardware, or like the DNA is the hardware, and then the app on the phone, this is the epigenetic program, the switching on and off of all the different genes at the right moments, especially during development of an egg, but it also happens throughout your life. And then again, the output during development is like the cell fate. So that's shown here. And so this is yet another visualization of this process. So here we have the DNA wound around these histone spools here. And then in addition, one other complication that happens is that these histone spools, these are essentially bundles of proteins. They contain these little tails hanging off the spool. And the tails those are the actual switches that allow the spools to be programmed. And what happens is different factors are flying around the cell, and they interact with those tails, and they can charge the tail, and they can cause these large-scale changes of thousands of spools to very compact conformations or very loose, spread-out conformations. And this is one of these programmers that charges one of the tails. And so again, the idea is that each spool has a chemical switch on it, and when it's switched on, the spools open up, and then the cell can then access the gene in between those spools. And that's the on state. Here's another example of the on state. With the orange region is accessible to the cell, and then it allows the gene to be turned on. Now, in the opposite case, when we're in the off state, here the histones or the spools condense to a very compact conformation. And in that case, the genes are no longer exposed to the cell. So that would be the off state. Here's that orange region that was necessary for the gene to be turned on is now hidden by all these spools that are tightly wound. Okay, so I wanted to try to demonstrate this with this physical mock-up I have here. And so here, so the idea is here, I have to give credit to my cat Willow, because Willow helped me put the stickers on this hose. So it took about 3 hours, and he was very good at keeping me company. And so the idea here is that you can imagine the hose as like a strand of DNA. And so here I have 4 different colors, like green for guanine, red for thymine, yellow for cytosine, and green and orange for adenine, for example. And if we tried to scale this model up, if the thickness of this hose were the same as the thickness of a molecule of DNA, there'd be about 10,000 miles of hose packed in this auditorium. So if you can imagine, going to Home Depot and doing an order, well, you'd probably have to do a huge order for 10,000 hoses, and then somehow have them flying around in this theater. That's what it would look like inside the cell. And then these garden hose spools are like the histones, the molecular spools, and they wind up the hose. And so the idea is, in some situations, the gene is turned on and the hoses are spread out, and you have this orange region would be like the gene activation region, and this would be exposed to the cell. So if I'm a factor, I could come flying around, and then I could, activate that gene and so forth. And then this here is like the switch. In this case, it's a lysine. And so to be more accurate, if I'm the factor, I wouldn't touch that region. I would just charge this part, the lysine. And then that would trigger the winding of these spools. So if I want to shut off the gene, I could charge that lysine, and then I could sort of wind these together. So we'll just kind of do the best we can, you know. Here. Yeah. This would be kind of a defect in the winding over there, I think. Let's try winding this here. So that could be a genetic disease. Okay, so then here. So anyway, you get the idea. So now this would be the off state. And so again, if you can imagine sort of thousands of those plastic spools, you know, hovering around in this theater and sort of unwinding and winding at just the right times and just the right moments, that's the epigenetic programming that's happening in all your cells right now at this moment. Okay. End of demo. And up in Los Alamos, we do a lot of different things. We do some computer modeling, and we do a lot in the wet lab as well. And so here's one of our computer models that we're running on the supercomputers up in Los Alamos. And this is a three-dimensional view of these spools. And this is magnified around 100 million times. And so we're running this on our new supercomputer called Trinity, which is coming out. Half of it's online now, and the rest is supposed to come online this summer. It currently ranks at number 6 in the world, and we're hoping to get close to number 1 when it's fully online. And so you can see these two spools are sort of closer together. And then what happens is when it's flooded with things like oxytocin, it triggers the spools to unwind, just like the garden hoses. And then the purple region would be the activation region that turns the stress response on. So we can watch again. We have 2 histone spools, and the DNA is wound around the histone spools. And then when these sort of ghost-like serotonin molecules are flying all around, it triggers different factors to come and charge the histones, and then this unwinding of the spools. This is not in real time, no. Okay, so one thing that's kind of spurred this whole explosion of epigenetics research in the last 5 years or so has been new DNA sequencing technology. So epigenetic effects have been known for a while, but in the last 5 years, there's sort of been a flurry of activity in the research community where everyone's trying to really hone in on how exactly do these epigenetic effects work at the atomistic level. And in addition, people are trying to figure out where and when do the epigenetic effects happen, what kinds of tissues, what stages during development, how many weeks after conception in the womb, how about in early childhood and so forth. And now with the new DNA sequencing technology, we're able to get DNA, whole genome sequences for different tissue types, for all kinds of different patients and so forth. And sort of this holy grail has now been reached where I think a while ago, people were sort of setting this grand challenge to do a whole genome sequence for $1,000, and that was reached about a year ago, I guess. And so now it's very commonplace to do full genomes. And in addition, you can use this technology to effectively get the epigenetic programs on top of the DNA. And sort of the killer app for this technology is stem cell programming. And so the idea here is that you begin with sort of a blank slate on your epigenetic program. You know, the egg is fertilized, and then that egg divides into 2, and those 2 divide into 2, and so on and so forth, until you get a ball of cells, and then inside the womb, and then that ball of cells changes as well, and different parts of the ball turn into arms and heads and eyes and everything. And it's often epigenetic switching that makes these decisions for the cell fate to take one cell into a blood cell and one cell into an eye cell and so forth. And so you can imagine it's very important to understand how that works. So when things go wrong, you know, if the epigenetics is not working right, you know, you may have an arm growing out of your eye or something like that. And that's something we don't want. And a long time ago, a famous person called Waddington drew this picture of something called the epigenetic landscape. And this is just another visualization of how we think the switching is happening. And the idea is that you begin with your fertilized egg, and then if you track single cells, so the egg will divide into two, and those two into four, and so forth. And if you follow one of those cells throughout its whole lifetime until the baby is born, for example, did that cell turn into a skin cell or a blood cell or what? And the idea is that at every point during development, decisions have to be made to go to the left or to the right. And then as more and more of those decisions are made, it specializes into the final adult cell. And so you could have a blood cell or a brain cell. And it's kind of analogous to a ski trail. And so suppose you're going skiing up at Santa Fe ski area, or if you happen to be at Heavenly in California, you have an enormous number of decisions to make. As you're skiing down, there's always a fork in the road or a fork in the ski trails where you have to go left or you have to go right. And a lot of these decisions need to be made. And so you may start up at the top here, and there's always these forks in the road. So you may want to go to a double black diamond if you're feeling more ambitious, or you could go take it easy on no grit and do an easier blue slope. And then suppose you may want to go for some wide open powder fields, or maybe hit the snowboard pipe and then do some gambling afterwards. In that case, you'd want to go to the Nevada side. And in addition, this analogy is also nice because it can show you how you can start at the bottom, go up to the top, and then make different decisions. So suppose you're in the parking lot here, and you want to end up in Nevada or somewhere else. You may take a gondola up, all the way up to the top, and then go down to the finish line here. And so the analogy here is that as the fertilized egg is developing, different decisions have to be made to get down to this landscape and to convert the cells into the different kinds of cells they need to be converted into, like a brain cell or a blood cell. So to get to 1 month, a lot of different decisions have to be made, and then 2 months and 14 months and so forth to get blood and brain cells and immune cells and heart cells and so on and so forth. And then in addition, there's also the concept of reprogramming. So a lot of epigenetics happens to take the egg all the way up to a baby. And then as far as stem cells go, the idea is to start with an adult skin cell, for example, and can we wipe the slate clean, erase all the epigenetic marks and switches, and then start with a blank slate and then program it into like a kidney cell or a heart cell or something like that. And so the idea there is you want to add some special protein mix to that cell to erase all the marks and get it all the way up to this blank slate, and then change it into some other kind of cell. And that's sort of the concept behind stem cell programming. And so why is there so much excitement about epigenetics? So again, the basic concept is that you have your DNA and genes, and then epigenetics is this layer of programming that can kind of turn things on and off. Well, it turns out that a lot of these switches happen during pregnancy. They happen, and also they happen throughout your whole life. So in pregnancy, and then as you're growing up, different kinds of epigenetic changes happen. And also, different chemicals in the environment can actually seriously alter the epigenetic switching and cause permanent changes to your genes. And then in addition, many of these changes can be permanent, so they can last for your whole life, and then they can also be passed down to up to 4 generations, so not only to the kids and the grandkids, but in some cases to the great-grandchildren. And then finally, one of the most intriguing things with epigenetics is that social interactions can also induce epigenetic changes. So things like stress during early childhood can actually alter the epigenetic program, and those effects can persist throughout your entire life. And I'll go into detail on one of those examples soon. And then finally, sort of the latest research that's coming out is there's a lot of positive effects that can come about with different epigenetic processes as well. So the main example I want to talk about today is about, it's in rodents and how mother rats nurture their young and the effect that has on epigenetic programming and on stress response. So if we go back to our question of nature versus nurture, remember that by nature we meant things that are genetically programmed or hardwired, and by nurture we mean things that are learned by experience. So as far as the stress response goes, the genetically hardwired version might be that some people are just born and they can't be rattled no matter what. They're just genetically unstressable, Whereas other people might be, easily startled by anything that goes bump in the night. And then as far as the nurture argument goes, there the idea might be if you grow up in a really stressful atmosphere, maybe that affects your ability to deal with stress later in life. Or likewise, maybe if you have a stressful job, that could also affect your stress response as well. And to discuss this example of the rats, the mommy rats nurturing their young and so forth, it's useful to think a little bit about the mechanism on how stress and stress response works in your body. And one analogy is that of just in baseball, a pitcher and a catcher. So in baseball, you have a pitcher that throws the ball and the catcher catches the ball. So there's the ball going and the catcher catches it. So that did not require the supercomputers at Los Alamos. Okay, so the analogy here is the ball is sort of like the stress hormone or cortisol. And so when a stressful event happens, you get a lot of cortisol that's excreted. And then if everything's working hunky-dory, you have this cortisol receptor that catches the cortisol hormone. And so if everything's working right, something stressful might happen, like someone took your parking space or something like that, and all this cortisol gets released and starts flooding your brain, but then you have the cortisol receptors. So the cortisol receptors realize that's happening, they catch all the cortisol, and you had that stressful event, but it's not going to kill you because someone took your parking space. Okay, so next, we can go into this in a little bit more detail. And so again, if you have the stressful event happen, you have the cortisol excreted, and you get cortisol receptor production, and then the receptors catch those cortisol molecules, and you have the happy response. Now, to talk about things going out of whack a little, I want to say a little bit about thermostats. And so, because we're going to talk about stress thermostats. So in your normal thermostat at home, you know that the thermostat sort of controls the temperature in your house. And so here, if you set your thermostat to 68 degrees, I set it to 78 degrees actually in my house, and it gets cold outside, then what happens is, or your cat is covered in snow also, so then what happens is your heater is supposed to kick in, and the heater turns on, but then when you get to, in my case, 78 degrees, then everything's fine. But, sorry, the clicker's going a little too fast. But what can happen is the thermostat can go out of whack sometimes, as many of you may have experienced, where it just keeps getting hotter and hotter and hotter, and then it's not working. So then Homer has to sit in his underwear and so forth. Okay, so the analogy here is that if there's a stressful event that happens, the stress response is very important to get right, and there's also a stress thermostat in your bodies that helps you deal with stress. And so the idea is that when a stressful event happens, cortisol is flooding your brain basically, but there may not be any cortisol receptors there. So the cortisol production may not be doing exactly what it's supposed to be doing. And in that case, you can end up with very high cortisol levels, and then you can end up with a very poor stress response. And so that's basically a primer on stress response and the biochemistry of it. So now I want to talk to you about this landmark study in epigenetics done by a great group in Montreal. And here they looked at mother rats licking and grooming their pups and the effect it has on DNA and the effect it has on stress response, not only during childhood, but all the way up through adulthood and into future generations. And so here they looked at two populations of rats. So in population number 1, the mothers were very good at licking and grooming their young. So they're kind of spoiling their young, driving them to soccer practice, giving them massages all the time. These are really nurturing mothers. Now, on the other hand, there was a second population where the mothers were not so good at licking and grooming their young and sort of neglected them a little bit. And what they did is they monitored the stress levels, and they also monitored the response to stress in terms of behavior. So they subjected the pups to different tests, like dropping them in water or facing them with a predator and so forth, and seeing do they really freak out or do they have a normal stress response. And then finally, they were able to look at the epigenetic switches on the DNA at the stress response gene. And so in population number 1, in this case, what would happen is the babies would get serotonin released, which is sort of the feel-good chemical when they were nurtured and so forth. In that case, they would look at the epigenetics, and here the stress response receptors were on. And then that would result in a normal level of stress receptors. So then when a stressful event did happen, they could respond very nicely to that stressful response. And then finally, in population number 2, in this case, where the mothers did not nurture their pups. Here, when you had a stressful event resulting in cortisol production, in that case, the receptors were turned off instead of being turned on. And so you didn't get the receptor production, and then you would have the angry or the poor stress response. And so the interesting thing is that when they looked at the DNA, they did see the marks coinciding with receptor off in one case and receptor on in the other. And then in addition, they also saw higher rates of diabetes and cancer in these animals. And finally, not only did the poor stress response persist through adulthood, but in successive generations of the rats. And so they would see these marks persist for many generations. So now we can talk a little bit about other studies. So there have been some interesting studies in mice where they've looked at effects on not only the children and grandchildren, but great-grandchildren. And this is by a group from Skinner and coworkers. And in these cases, they looked at different effects such as jet fuel and pesticides, bisphenol A, and bisphenol A. And so in this case, they saw that the pups had fewer eggs and also a large number of ovarian cysts, and these persisted all the way through to the great-grandchildren. So it's interesting that these epigenetic effects, once these switches are switched, it persists for many generations. And then in humans, there have been some interesting studies done. So one has shown that child abuse alters the epigenetic profile in the postmortem brain. Another one showed that stress on the mother caused by violence of a partner caused epigenetics on the same cortisol receptor in the blood of adolescents. And they've also seen marks in populations with higher rates of suicide. And also there's a lot of exciting research going on in autism now where they see this oxytocin receptor gene turned off epigenetically. And so right now there's not a whole lot of smoking guns, I would say, in human studies, but what they're doing is they're just finding a lot of correlations with epigenetic marks on the DNA and then these different kinds of things happening. Now, as far as positive effects go, some of the latest research that's being done are looking at more positive things. So there's a group in Sweden looking at exercise. And I was at a conference a few years ago, and the one person showed a poster where they saw from a single workout, they could see epigenetic marks at these fat storage cell sites. And then in more long term, they looked at a population with 6 months of exercise, and they could see that these fat epigenetic circuits were reprogrammed. And then some very exciting studies are showing recovery of lost memory in mice. And in these cases, they're starting to come up with epigenetic drugs that kind of freeze these epigenetic programmers. And so kind of the state of the art now, and as far as epigenetic drugs go, is that they're more sort of almost analogous to broad spectrum antibiotics, where they're sort of hitting epigenetic factors in all of the cells in your body and then hoping that it does some good. And in a lot of cases it is. And sort of the future is about making things more specific to specific tissues and so forth. Okay, so now I want to just say a few words about cancer. This is one of the active areas of research in epigenetics and drug design for epigenetic effects. And so if we can go back to our smartphone metaphor. Now remember before we said that the DNA is kind of like the hardware, and then the epigenetic program is kind of like the app, and then the cell fate was kind of like the output of that app. Well, if we take the analogy a little bit further, we can talk about these epigenetic factors are sort of the programmers that program the app, and that the epigenetic marks or switching states are kind of like the settings in your smartphone. And so right now, the cancer drugs that are out there now sort of freeze all the settings in all the cells, and they hope it does some good. You know, it's better than something like chemotherapy that's sort of trying to kill all the cells. In this case, you're not killing the cell, you're just freezing the epigenetic programmers and freezing those settings. So it's kind of a more subtle effect than your normal chemotherapy. And so when we think about cancer, the idea is that in sort of a big picture view, the idea is that you have tumor suppressor genes and then you have oncogenes. And so the tumor suppressor genes help suppress the tumor growth, and then the oncogenes promote the tumor growth. And then in a healthy individual, you always have this balance. So we all have tumor suppressor genes and we all have oncogenes, but there's always got to be a balance between the two. And then when things go out of balance, that's when you can get cancer. And so we can go back to our ski trail metaphor, and like I was saying before, that when you start a cell from sort of an embryo state, it has to divide and differentiate, and then it can turn into a blood cell or a brain cell or an immune cell or a heart cell. Well, in addition, it could also turn into a tumor cell as well. And so it's important to understand, you know, how this process worked and how epigenetics controls the change of these cells. And so ideally, what the goal here is to start with a cancer cell and then reprogram it. So wipe out all its epigenetic marks and take it to the top of the hill, and then let it be programmed into a cell that can actually be beneficial to your body rather than taking away from you. And so as far as the epigenetics goes, in just normal cells, as I was saying before, you can imagine it as these, you know, thousands of these plastic spools hovering in the auditorium and turning things on and off and so forth. Well, it's going to turn the tumor suppressor genes on, and you're going to have your oncogenes turned off. And then in addition, there's sort of a third class of genes that are important for cancer, and these I just call sort of DNA chaos genes, where they cause kind of swapping of different parts of genes from one spot to another. And in those cases, you also want those turned off in a normal cell. But in cancer cells, what happens is that the tumor suppressors, the things that are supposed to suppress the tumors, are actually off, resulting in tumor growth. And then the oncogenes are turned on, also resulting in tumor growth. And then there are also other kinds of situations where these so-called DNA chaos genes are also turned on, causing, you know, widespread swapping of genetic elements and so forth. And some other kinds of programming, epigenetic programming errors that happen is sometimes you can have just too many epigenetic programmers out there. And this happens in breast cancer, in leukemia, in prostate cancer, and in liver cancer. In addition, you can have the wrong programming team present or the wrong kinds of factors present. So that would be, for example, if you had an Android phone, but you had like your team of Mac programmers trying to debug it, and they're not doing too well. And then finally, you can have a situation where your spools are sort of at the wrong place on the genome, and so the spools are not where they should be. Those are other kinds of epigenetic errors that can affect cancer. And then as far as epigenetic cancer drugs go, as I mentioned, the traditional cancer drug is to sort of kill the entire cell for both cancerous and non-cancerous cells. And that's why in chemotherapy there'll be really nasty side effects that you get, and it practically kills you when you have to go through chemotherapy. And the idea in epigenetics is to block all the programmers in both cancerous and non-cancerous cells, but at least it halts, for example, the DNA chaos genes from being turned on and so forth. And then the ultimate goal is to sort of reprogram these cells rather than kill the cells. And right now, there are a few epigenetic drugs on the market. These are more broad spectrum. And I just read an article, they're expecting the epigenetic drug market to grow to like 30 billion by the year 2020, I think. So it's a really exploding field as far as drug development goes. Okay, so that was sort of a little blurb on cancer and epigenetics. And lastly, I want to talk a little bit about what we're doing up in Los Alamos. And so here, what we're trying to focus on is a huge outstanding question in epigenetics. So I told you about the spools, these plastic garden hose spools floating around in the theater and the hose winding and unwinding. And those spools, they have to sort of wind and unwind at just the right spot in just the right moment. Now, the big question is, how do they know when to unwind and when to wind? How do they know how to do it right at the right time and also right at the right place? This, no one really has any good idea. And it turns out that somehow the factors come and turn them on in the right spots at the right moments. And there's some kind of recruiting mechanism that must happen that can sense what's happening. So some kind of communication has to happen where somehow the cell, the information of the environment of the cell, is speaking to these spools, telling them what's going on in the cell. And the question is, how does that communication occur? And that's what we're working on in Los Alamos. And we think the answer is RNA. And RNA is sort of this cousin of DNA. And so our group is focused on understanding how the RNA can talk to the spools and trigger these different effects. And so here's just sort of a crude cartoon of what we think is happening. And again, we have these spools that the DNA is wound around. And then we have the factors that do the switching. And somehow the spools have to talk to the factors. And then what we think is happening is that these RNA molecules are being synthesized from the DNA, and then they're folding into specific shapes and then recruiting these epigenetic factors to the DNA, and then they can bind and make their switching marks on the DNA or on the histone spools. And just to give you a one-slide summary of what RNA is and why it's important, there are like thousands of people working on this for like 50 years. But so RNA is kind of like DNA's molecular cousin, and it's kind of like it's kind of like a disgruntled teenager. I'm not sure how to say it. It's like it's never appreciated, you know. And so DNA, everyone knows what DNA is. It has the blueprint of your genome, and it stores all the information for your body, you know, on that molecule. And then what happens is the DNA is converted into proteins, and the proteins do all the work. They make up all the structures, and they do all the chemistry and so forth. So I can be talking to you right now. And for a long time, people thought RNA was just this sort of go-between. It would just like read what's on the DNA and then turn it into proteins, and that's what RNA did. But in the last 10 years or so, it's been found that RNA actually can stand on its own and really do its own thing. And so it's been found that RNA can do lots of great things in the cells. It can do chemistry, it can make structures, and it can do all kinds of different things. And one thing that it does a lot is epigenetic switching and epigenetic programming. And people think it may be the sort of missing link to recruit those programming factors to the DNA. And so when you have your strand of RNA, it has a sequence just like DNA has a sequence. And then one thing about RNA, though, is it can fold up into more complicated shapes. So DNA pretty much just folds up into this double helix, the famous double helix. And that can bend around a little bit, but it's basically a double helix. But RNA can do lots of more folds, so it can make these cloverleaf shapes, and it can make all kinds of different shapes that have helices in them, but they're kind of warped and twisted and put together in different ways and so forth. And then in 3D, this is what a little piece of RNA looks like that does something. And so what we're doing in Los Alamos is trying to map out the structures of some of these epigenetic RNAs. And so it's been found that a whole new class of RNAs exists that's responsible for epigenetic effects, and this is called long non-coding RNAs. And sort of the coding RNAs are the ones that are these go-betweens just going from the DNA and making protein, DNA making protein, DNA making protein. But these non-coding RNAs are what people used to think were just junk, or they used to call it junk DNA and so forth. But it actually, the junk actually does a lot of important stuff. And many people believe that these RNAs are really critical for almost every different kind of epigenetic effect. And so what we're doing is we're trying to map out what these RNAs look like. And sort of an outstanding question in this new field of long RNAs is that, do these RNAs have any structure at all? Are they just sort of like floppy, like wound up and tangled in a bowl of spaghetti? Or are they really organized and highly structured? And can they sense what's happening in the cell and then talk to these spools? And so what we're finding is that they do seem to have structure. And we've looked at about 4 of these RNAs now. This is the first one we looked at, called the steroid receptor RNA activator. And this is an RNA that's responsible for upregulating the estrogen response in human. And it also interacts with many other hormones. And so we mapped the structure of this. And this was done by Irina Novikova and Scott Henley in the lab, also with some help from Ashutosh Dharup and Serdal Kermizialtan, and they were able to find that this particular RNA had all kinds of structures, 20-some helices, making lots of intricate interactions and so forth. And so this isn't, this is just sort of the first step in figuring out what these RNAs do, but it's a very interesting result. And so sort of the cutting edge of epigenetics, one of the key questions is sort of, how does the DNA know what's going on in the cell? And how do these spools know when to wind and unwind? And we think the answer may lie in RNA. And these RNAs have been linked to a lot of different things, like Alzheimer's and autism and different kinds of important phenomena. And so one kind of interesting idea, in these Santa Fe lectures, we're supposed to present one controversial idea. And so this idea is that this is what I believe. And so they're sort of like, it's hard to explain, but they're kind of RNA people, and then they're people who are not RNA people. And so, and so the RNA people tend to be diehard pro-RNA, like no matter what. And so I think many people in the RNA community believe that the cell may be teeming with these long RNAs. Just everywhere you look, there are these long RNAs doing all kinds of stuff, and that they may be highly, highly structured. And the controversy now is, are they just flopping around like spaghetti? Or are they really very intricately structured? And we believe that maybe there's many of them that are intricately structured. And there are a lot of studies showing that there is many of these non-coding RNAs as there are proteins. So that's interesting. And I think each year there's more and more evidence coming out that, yes, these RNAs are really important and really important in epigenetics. And so as far as the future of epigenetics goes, we're sort of just at the baby stages now in terms of actual mechanism and figuring out what's going on atomistically and what causes what and so forth. But the good thing is that there's just tons of data coming out each year, and with the cheap new sequencing technology, there's just going to be more and more data, especially the sort of personalized data for different patients and different cell types, lots of human studies, and the drug companies are really pouring money into this problem as well, and they're very interested in this as well. And so with all this progress, it kind of brings us back to our beginning question we opened is, how do we know we're in love? How do you know you're in love? And we don't really know the answer to that now, but maybe in the future, when your kids ask you, how do you know you're in love? You might be able to answer, well, it's simple. You know, when histone 3 is chemically modified. And so that's how you know you're in love. Thank you.